Okay, we're getting started here. Very, very pleased, very happy to have Chris Wells giving a talk today as for his PhD. Chris came to us from the University of New Hampshire, where he did an undergraduate degree, finishing in 2009, and then stayed on there for his master's, and finished that, I think, in 2012, 13, okay. And then started here in our program in 2014. He got interested in sea anemones early on as an undergraduate, working on a research project with Larry Harris at the University of New Hampshire, looking at what appeared to be a invasive species of anemone that he then continued work on for his master's, and all of a sudden it disappeared. And he spent some time trying to figure out why, and I believe it was a combination of cold temperatures and the fact that this particular invasion was all one clone. So that was a very interesting project, but it just, it ended. So he took on some other projects during his graduate work, and he's been on a number of publications from those projects, including one going to work in Belize at the Smithsonian Station, working with one of their researchers. Then he came here. No, actually, there was one other step in between. He applied here, and I said no. That year, that year I didn't have fun. My funding was ending. I think I didn't feel I had room for more students, but I changed my mind the next year. So I said yes that time. And very glad I did. So when Chris came here, he continued his interest in sea anemones, their ecology, and various aspects of their biology. When he was deciding, which we'll hear about, when he was deciding what was going to become the pieces of his dissertation, it was difficult because he actually had probably twice as many things as he's going to talk about today that could have gone into the dissertation that are going to be papers and so on, ongoing projects. But he picked three good ones, including one that's already published, and he's going to tell us about that today.
Thanks, Ken. Thank you guys for coming. As you guys are probably all aware, I am interested in sea anemones, but more broadly Hexacorallians, so corals and other things as well. And I did nearly all my work up in the San Juan Archipelago. So I'm going to be talking about some of the work I did over the past 5 years. Before we get into the actual bulk of the talk, we're going to talk about what is a hexacorallian. Hexacorallians are a group of benthic invertebrates. They are attached to the bottom their whole life. There are 4 major orders: the scleractinians, or the corals, the actinarians, or the anemones, zoantharians, and the corallivores. Around here, we have representatives of all of them. These are all pictures taken Saturday.
Wow.
Yeah. And all of them fall within the phylum Cnidaria, which is a unifying characteristic of that group, is that they all have stinging mitocysts. I think most, if not all of us, are aware of the importance of corals in providing habitat for a myriad of animals. In the tropics, coral is one of the most conspicuous and dominant suspension feeders in their ecosystem. But less people probably think about temperate hexacorallians, but they also can be quite dominant. In particular, the dominating group in the colder waters is the sea anemones. The Pacific Northwest has one of the most diverse assemblages of hexacorallians, temperate hexacorallians in the world. And this diversity allows for an anemone biologist such as myself to explore a broad range of ecological questions without having to stray away from my favorite group. Just to highlight sort of the diversity in this picture, there are 4 different species of sea anemone and then 1 coral. So 1, 2, 3, 4 anemones and then a little coral, or one and only coral here, at least in the shallows. Right, so this is sort of the overview of my dissertation, but also this presentation. I'm going to start off looking at the individual effects of a particular marking technique, then go on to sort of the population, the diet of a population, and then sort of expand to the patterns that you can determine based on looking at multiple different species. And each one of these chapters will sort of tiptoe through and sort of get broader and broader. To start, let's talk about marking anemones. One of my sort of anemone loves is Metridium parsimon. It's this really large white sea anemone. It's about 5 feet tall, or 1.8 meters if you're into the metrics. It's almost exclusively subtidal. It's a competitive dominant in its environment, particularly in subtidal ledges and undersides of floats, which you'll see later. And compared to a lot of the other species, here is relatively well studied. That being said, its ecology it's not so well understood. We don't know how much it eats. We don't know, we know very little bit of its development, other than it has a planula stage and it may or may not feed. We don't know how long it takes to get to full size. And a lot of these sorts of questions and these pieces of information that we could glean from this animal, we need to be able to track individuals over time. And while that may seem easy, it's a little bit more nuanced than that. And so I'm going to talk a lot about marking anemones and sort of some recommendations for what I would recommend you do. Tracking animals isn't a new thing. I think almost all of you are probably familiar with bee tags, super glue on the back of a bee, a number. Then you can watch bees do their thing, go pollinate, interact with one another. They've expanded that method to lizards. I just wanted to include this because it's kind of cute. Looks like a lizard's wearing a hat. But once you get into the aquatic environment, then you have to start dealing with hydrodynamics. And so that's not to say that it hasn't been done. For example, you can mount a depth logger to measure vertical migrations. This is one of Peter Ward's papers looking at the nautilus. You can, for completely sessile species like corals and algae, you can put a tag nearby and that'll suffice. That's a lot of ecologists use these. They're swine ear tags that they just cement down to the bottom. But the trouble is with sea anemones is they're modal. 1, they're soft body, but 2, they're modal. And so they've been a tough bug to tag. So unlike a lot of invertebrates, they don't have hard parts. Like a lot of invertebrates, they do move. And they also have amazing regenerative capabilities. And so the first sort of iteration of this was running fishing line through them, sort of sewing a tag on them, but they can squeeze that out of their body and then it falls off and you can't tell one from the other again. Not to mention the whole time they're moving around in these groups. So like, while this may look like a static group, each of these is individually moving, and as one moves, bugs the other, this one moves, and so forth, so on and so forth. Ken, in the 70s, came up with a method for marking sea anemones using neutral red paint. So neutral red dye, mix it up, paint it on the anemones, and you can number them, you can draw symbols on them, and then you can come back to individuals in the intertidal. The trouble is they have to be out of water for a portion of that, and that works great for intertidal anemones, but when you try to work with a subtidal anemone like Metridium, you need to take them out of the water. And even if you are an expert anemone wrangler and don't damage the pedal disc at all, it takes several hours for them to reattach. And so you need to be able to set up some sort of contraption to hold the anemone to a vertical surface for several hours in high flow environments like the San Juans. And so that was just not something that I was interested in doing while diving. And so I tried to, Ken and I came up with a method of marking Metridium using the method he had used, but instead of painting it on, we were hypodermically injecting them with stain. So we tried 3 different stains, neutral red, methylene blue, and fluorescein. All of these stains have been used in some manner with invertebrates, and so we thought, should work. And then, so that was one of our questions was, do these work? Do they affect growth? Do they affect survival? And then secondly, how long do they last? So let's get to the pretty pictures. If you stain anemones, you can make them really brilliantly colored. And so these are anemones from the experiment that I did. On the top left, you have a seawater injected Metridium farcimen. And if you don't believe me, I'll show pictures later, but that looks like a normal Metridium farcimen. On top right, you have methylene blue, bottom left, neutral red, bottom right, fluorescein. And while fluorescein looks amazing, it's lethal. After several days, the sea anemone starts just eating itself from the inside out. I don't think it's eating itself. I guess it's just sort of disintegrating. It's a pretty horrid thing. Don't recommend. It's surprising, though, because fluorescein's used quite frequently in vertebrate zoology. We use it to visualize flow in bivalves, in brachiopods, in, I think, nearly every marine biologist at some point in their life has seen a YouTube video of a sponge biologist squirting some fluorescein on the outside. It goes through the ostia and it comes out the oscula in this really magnificent glow-in-the-dark yellow color. Don't do that to sea anemones. Also, we use it to visualize scratches on our eyes. So it's got a range of applications, but not with sea anemones. The other 2 markers, the methylene blue and neutral red, do not affect growth. So if we're looking at the lab study here, X on the X axis is time, and on the Y axis you have pedal disc diameter, aka size of the anemone. Each of those dots is 21 different individuals and standard error. You can see from this figure that there's a 2-week, in the first 2 weeks, there's a dip. So I was measuring every week. There's a dip, and this is probably some healing from me harvesting / me injecting them with a 16-gauge needle. But after that, they all grow, and there's actually no significant difference between either methylene blue or neutral red and seawater injected anemones. For the methylene blue, the reason we stopped this study, the lab study, is that after about 45 days, you really can't tell the difference between the methylene blue anemone and a seawater injected anemone. The neutral red, on the other hand, keeps going. So this is after a year. This is an anemone. This pink guy right here is an anemone that's been marked with neutral red about a year ago, and you can see that you can still tell the difference. And some of these anemones, I've been able to visit after 3.5 years, and you can still tell if you squint really hard and tilt your head just right that there's some pink anemones in the clusters. And so if you do this technique along with a swine tag next to an aggregation, you can mark a bunch of different anemones on a wall and measure growth, for example, which is something that I'm not talking about, but trying to figure out how old these anemones get. The sort of punchline is probably takes about 15 to 20 years to get to maximum size. So it takes a while for these suckers to get to the size 20 centimeter diameter animals. That's not the only thing you can do with this labeling technique. You can, if you wanted, look at how tissue regenerates over time. You amputate a piece of Metridium and you inject it with some neutral red and then see how that diffuses across an anemone as it regenerates, or you could take a tissue, neutral red tissue from one anemone and graft it on another and see how the cells interact in some way. There's all sorts of applications, and we could chat about them for longer, but one of the other sort of clear results of this that I think that people have been banging the drum for a while is that you need to figure out, if you're going to mark an animal in whatever manner, you need to make sure that that marking doesn't affect their behavior or their survival or their growth, depending on what you're sort of looking at. And so neutral red and methylene blue work great. And I would recommend methylene blue for short-term studies, neutral red for long-term. And if you have, if it's short-term, you can also use both in combination for some cool tie-dye type anemones. I just want to point out that I didn't end up using this method in any of the other chapters, so it's sort of disjunct from the rest of the dissertation, but there are other places that I've used this method in my research. It does work, just not in the next two chapters. And you can find this too, it's already published in PLOS One. So just to summarize, neutral red and methylene blue are effective stains. They don't significantly affect growth or survival, although fluorescein does. And methylene blue lasts about 6 weeks, while neutral red lasts at least a year. Okay, chapter 2. So let's switch gears from marking anemones to figuring out their diets. This video is from underneath Centennial floating dock. If you've ever been to FHL, there's a large research vessel that floats right next to it, are completely covered in Metridium farcimum. And this is where I sourced the anemones as well, but just show how dominant these guys underneath the floats. You can't see nearly anything else. So we've been talking a lot about Metridium farcimum, sort of how they mark. Now I want to sort of broaden that out to populations of Metridium farcimum. In particular, what are their diets? And I'm focusing on this population in particular. You may remember that Metridium farcimum is a suspension feeder, specializes on zooplankton. But zooplankton is a difficult group to work with, and I wanted to try a different method of analyzing the data and compare that to sort of traditional gut content methods. So in this chapter, I'm going to discuss the diet of Metridium, especially in comparison to concurrent plankton sampling, and then compare my findings using DNA metabarcoding to some more traditional gut content techniques. So on hard bottom, so just to give you a heads up on what this picture is, it's a field of sea cucumbers, probably all about this teeny, and then in the background you've got some Metridium farcimum, all suspension feeders. So on hard bottom surfaces, substratum feeders dominate the benthos, and these suspension feeders have been shown to have major effects on the communities above them, the plankton. For example, acidian and polychaete filtration rates have been calculated to be able to deplete the phytoplankton within the first 30 centimeters of the benthos. But plankton aren't passive particles. They do have some defenses for not getting eaten. They can be toxic. They can have inducible defenses. For example, Daphnia and their helmets and spines. And they can have all sorts of capabilities of escaping or detecting predators from afar. And these abilities to escape predation alongside preferential capturing a particularly palatable prey is called prey selection. And that's what we're going to be talking about here, is a lot about prey selection, are Metridium selecting their prey. And that doesn't mean positively or negatively, just but generally. Studies like this on prey selectivity allow for a better understanding of the effects of predators on their ecosystem, prey capturing mechanisms, and dietary niche partitioning. That's a porcelain crab. Doesn't have inducible defenses, but does have some serious defenses nonetheless. Unfortunately, quantifying the diet of a planktivorous animal is difficult. Plankton are microscopic, very difficult to distinguish as larvae, may not have diagnostic characteristics of larvae, and are digested quickly and at variable rates. Soft-bodied animals generally are digested more quickly than hard-bodied animals. And this makes marine plankton difficult to identify in gut contents past the class or order using traditional identification techniques. And for those who aren't schooled in the traditional identification techniques of gut content analysis in sea anemones, what you do is you collect a sea anemone off the wall like this, bring it into the lab, not safe for work picture, bisect the sea anemone longitudinally, scrape out the internals to get at whatever is in amongst all of their tissue, and then move that tissue into a clean container so it looks pretty, with copious amounts of rinsing of ethanol because that breaks up the mucus, and then identify everything that's in there. A large majority of that material is going to be anemone gut, anemone reproductive stuff, not particularly organs because they don't have them, but reproductive tissues. But you can see here, a little gamete amphipod. This is a time-consuming process, but you can get a lot of information, especially on life history, sizes of sort of critters that you're looking at. I went with a different technique, a molecular technique, where we called DNA metabarcoding, which makes the process a lot faster and a lot more easy. And while I think I have the taxonomic chops, you don't need to have taxonomic chops in order to do it. So I'll sort of go through sort of the basic sort of explanation of what metabarcoding is. So what you do is you collect a sample, whatever that may be, in this case, the gut contents of Metridium farcimen. You grind that up into a paste, spin it in a centrifuge to get a pellet, extract the DNA from that, send that DNA off to a sequencer, and then you go and match those sequences against already published sequences or your own secret stash of sequences, and then publish that secret stash of sequences along with your paper to identify what you have there. And that works for a good portion of the stuff. I'll say that about 25% of my the operational taxonomic units they're sort of like species that identified to a species level. So it's about 25%, quarter of them. The rest are to a broader category, genus, family, order, a lot of them phylum, or just animal. And I have, I don't know, like 72 animal species. But you can still track communities. Oh, it's in this group, it's not in that group. Still interesting information. So what you do get from this compared to the traditional methods is you get the ability to identify things that you would never be able to identify from traditional methods. For example, if you have a freckle of a copepod in there and it's got some DNA associated with it, you can identify it if it's got a barcode to species. I can't identify freckles of copepods personally, but with metabarcoding, you can. And so what we did with this information is we compared the gut contents of sea anemones, Metridium farcimen, with nearby plankton samples. So we had two questions for this chapter. One was, does Metridium farcimen have a selective diet? Is it preferring certain things, avoiding certain things? Can certain things escape predation? And then how does DNA metabarcoding compare to traditional gut content analyses? So I did this work up in the San Juan Islands. For those that may not be familiar with the West Coast, I think everyone here is, but hopefully there's some people online that maybe haven't thought too much about the West Coast yet. We're working up in Northwest Washington in the San Juan Archipelago, which is in the Salish Sea. In particular, for this chapter, I was working Harbor, which is this little embayment right here in the San Juan Island. I'm going to blow that up a little bit. These were the 3 field sites where I collected plankton. And then, so all those 3 red dots are plankton samples. I collected with 2 different size meshes. One was an 80 micron mesh, one was a 330 micron mesh. 80 collects pretty much, well, not everything, but a whole lot of stuff. And then 330 micron filters out a lot of the teeny stuff. And then compare that to the difficult to see but yellow dot behind the population of Friday Harbor Lab Metridium to see how do these compare. So let's jump into the results here. On the x-axis, you have the different communities I sampled from. You have gut contents, 80 micron filtered plankton, and 330 micron filtered plankton. And on the y, you have percentage sequences. For those that aren't so invertebrate oriented, you don't need to know much about these groups other than to just sort of compare the relative proportions of each of these. And hopefully you can see that the Metridium gut contents is much more similar to the 80 micron filtered plankton. But if you, and I want to point out, just because it's the next thing we're going to look at, is that these categories, the arthropods, are relatively similar in proportion of their diet, but this is at a phylum level. When you open it up to sort of a class level, on the x-axis you have the classes, on the y percent of sequences, and then blue is Metridium, red is filtered plankton. You can see that there is quite a substantial difference between these things. And as you go from class to order to family to genus, this just continues to broaden the gap. That would be interesting enough, but there's this really interesting group that probably should, at least initially in my mind, shouldn't have been there. Insects. As far of the insects that we found, none of them were particularly associated with the marine environment. They're just, I think 8% of that bar, or not 8% of that bar, but 8% of their diet was actually ants. And that, there's a lot of reasons why that could be. I think that that's accurate, but I think that's probably an artifact of timing. My guess is that this was during a mating flight of that particular species. But it highlights the fact that during certain parts of the year, these floating communities on docks have a significant portion, 10% of their diet is terrestrially derived foods, which is really cool. The other thing to point out is that the holoplankton, the hexanoplei and the branchiopods, although there are barnacles in here as well, seem to be able to avoid predation or are not preferred a little bit more. So you can already see there's some sort of prey selectivity going on here. And to look more at that prey selectivity, don't particularly delve too deep into each of these, but sort of pull out and look at all of these taxa. None of these taxa were significantly more abundant in the Metridium guts. There were some, many were underrepresented. And what this means is that if there aren't any that are overrepresented, that means that there aren't any zooplankton that are being preferred. There's nothing getting pulled out of the plankton at a higher rate than is to be expected. But there are things that are seemingly avoiding predation statistically significantly. And all of these are, some of them 121-fold, others only 2, but lots of different hexanoplei, some rotifers. And there's probably multiple reasons for this. I would expect that the rotifers are probably too small for Metridium to be able to detect or effectively consume, while things like hexanoplei may be able to escape predation because they are relatively good swimmers compared to other zooplankters. If we look at sort of overall composition of the gut contents, there are significant differences between the plankton communities and the Metridium gut contents. If you don't, or if this is the first time you've seen an NMDS plot, or you've never sort of been able to read them quite well, hopefully I can explain to you. Each dot is a sample. Each box or sort of shape is a treatment. And if the closer the dots or the boxes are to one another, the more similar they are, or they're likely more similar to one another. And the emphasis is on likely, because this is squishing an 18-dimensional space into 2 dimensions. And so I can't imagine it. I don't want you to try to imagine it, but suffice to say that there can be differences even if it doesn't look like there are. Also, the scale, the axes is arbitrary. I could rotate this 80 degrees if I wanted, and it would say the same exact thing. So don't be hung up by X and Y axes. And what you should see is that the 80 micron filtered plankton, which we've already talked about, is more similar to the Metridium farcimen gut contents, but they are different. The statistical analysis I use, the Permanova, indicates that they are two separate communities. Another interesting thing to discuss is that the Metridium farcimen gut contents are much more dispersed than either of the filtered plankton. And so if you were to verify this down, so it's just 3 samples of any of the Metridium farcimen, you're likely to get a larger spread than either of the plankton. And so I think that's an indication of the fact that Metridium farcimen is sampling over hours. So it's a snapshot. It's not a snapshot like the plankton is. It's sampling over hours, which is an interesting result. Too far. Spoilers. So lastly, I'd like to compare the different studies that have looked at Metridium farcimen. The first 2 studies I'm going to show you have looked at diet composition using traditional methods, and then I'll show you my results, although you got to see it a little bit. You don't particularly need to know about each of these classes, but it's important to see just how many of these show up with some sort of color. Red meaning that they're particularly abundant, blue meaning that they're particularly not abundant, but present, and then gray is in between. You can see that there's 7 different classes that Purcell found in California. Ken did a study in, is it South Puget Sound? Looking at, and he also found 7 different classes, slightly different results. But then when you look at metabarcoding, this study, there's 26 different classes, and so it's like a 4-fold increase. And so metabarcoding allows for that higher taxonomic resolution, allows you to identify the freckles that you wouldn't otherwise be able to identify in your sort of traditional techniques. That being said, it lacks the ability for you to look at sort of life history stuff. For example, I don't know what stage of hexanoplei that I was looking at, but Ken in his paper can tell me the size of it and can tell me whether it was a nauplius or whether it was an adult or whether it was somewhere in between. And so there, you do lose some resolution elsewhere in order to increase taxonomic resolution. It depends on what your goal is. So to summarize chapter 2, the gut contents of anemones are more dispersed than the plankton. This is likely because of the way Metridium is consuming over time, whereas plankton is a snapshot. There are no statistically overrepresented species, but there are a bunch of underrepresented species, and so there is prey selection happening. There is a surprisingly high level of terrestrial input, 10%, and that may just be for this community. I assume that a Metridium farcimen at 8 meters depth is not eating ants, but it does highlight the sort of connectivity between floating dock communities and terrestrial communities, for example. And then metabarcoding finds many more taxa, but has its drawbacks. All right, onwards to chapter 3. So as I've stated before, just a wonderful picture at the edge of this guy. Wow, that site. The whole reason I did the chapter sidebar is so that I could go dive at this site. Amazingly gorgeous. Anyways, Hexacorallians are, we've already talked about this. Hexacorallians are prominent passive suspension feeders in most benthic ecosystems. Temperate zone hexacorallians, like their tropical relatives, are the dominant space occupiers, and some facilitate the recruitment and survival of things in their understory. And the San Juan Archipelago is one of the most diverse areas for temperate zone hexacorallians in the world, with 18 hard bottom species, not to mention the soft bottom species, not to mention the undescribed species, of which there are several. The ecology of some of these species is relatively well known. For example, Anthoplura, Elegantissima, and Metridium farcimen have had quite a few studies on them. Whereas Epizoanthus and Crypronopsis have had, to the best of my knowledge, nothing has been done with them other than, look, they exist here. One associates with a shrimp. We don't know what's going on. And so I wanted to look, and because I had this unique opportunity of being able to study a bunch of different anemones all at one time, I was hoping to be able to see if there was any sort of pattern across Hexacorallia and how density was affected by several different factors. So let's tiptoe through some of those factors. So many researchers have shown that different assemblages develop on surfaces of different orientations. The reason for such differences may involve light, predation, grazing, larval behavior, and water flow. And here you can see two shallow substrata. One is a horizontal surface, one is a vertical surface. The horizontal surface is very typically dominated by upright algae, whereas the vertical surface is dominated by invertebrates and algal crusts. And that's what you see all around the San Juans. Additionally, depth is a major structure of populations. The clearest difference between shallow communities and deeper communities is the influence of light. And so depth and light are nearly synonymous in the marine community. In shallow environments, there's sufficient light to sustain large stands of upright algae, but as you get deeper, this falls precipitously off and you end up having an invertebrate-dominated community. I don't have it here, but most of the invertebrates in the shallows are on vertical surfaces, and then it sort of opens up to all surfaces as you get deeper. Another factor that has some clear impacts on community composition is flow. Flow brings oxygen, it brings nutrients, it removes waste, it transports larvae, but there's a cost to being in a high flow environment. Your feeding apparatus, if you will, can deform, and if it's exceedingly high, they can be ripped off the bottom and transported potentially to somewhere that's less than desirable. But many Hexacorallians can modify their flow. They can just close up and not deal with it, or they can live in aggregations which can reduce the flow, and then which then may increase prey capture. This is shown. It's been shown particularly in coral reefs with coral heads. They really can impact this. Predation and temperature can also have some major impacts on benthic invertebrates. And for example, Anthoplura elegantissima, its lower bound is of its distribution depth-wise is controlled by the presence of, or has been shown to be controlled by the presence of sea stars and nudibranchs that eat them. This is Elydia papulosa, unless its name has changed recently, eating a poor little Anthoplura. But if you transport Anthoplura to the subtidal and cage them so that they're not getting attacked, they grow better than in the intertidal. So there's a clear sort of indication that they might prefer to live there, but if they live there, they're toast. And so I wanted to look at, I think I've just talked about 6 factors now: flow, light, depth, substratum slope, temperature, and predation on the distribution of the 7 most common hard-bottom Hexacorallians. And so here's sort of chapter 3's questions and hypotheses. I think I just said the first one, but all 6 of these factors have been looked at separately, some paired, but their interactions and which of these is most important to some of these species hasn't really been looked at. Additionally, I'd like to be able to look at sort of more general patterns across the Hexacorallians, and having this diverse group of animals across 3 of the 4 different orders of Hexacorallia allows me to look a little bit more at some generalized patterns. So I worked once again in the San Juan Archipelago, but not for Freddie Harbor this time. A little bit in Freddie Harbor, but most all over the place. And so I picked 12 different sites around the islands. This is about, what do you say, an hour boat ride from Freddie Harbor. So it's quite a distance in between. I forgot to put a scale there. But I chose these 12 sites with several different criteria, one being that across the 12 sites, we had a range of flows experienced, that it reached 40 meters depth in a reasonable swimming distance, because I don't want to swim more than 300 meters in a dive. Also, I don't have the air to do that. And then had a variety of horizontal, sloping, and vertical surfaces at all depths. So it wasn't just one big wall and then it became flat type of thing. It was sort of a little bit more rugose, if you will. And so I took lots and lots and lots of pictures from 38 meters below mean lower low water and 4 meters above it. 2 photographs every half meter depth, sometimes multiple transects at a site. And this allowed me to cover the intertidal community, the shallow algal-dominated community, and then the deeper invertebrate-dominated community. Here are 6 of the 5,766 photographs I took, some of which were Kens, so probably more like 3,000 Kens. And each of these photographs is from about the same depth, 6 different sites. You can see that there's quite a big difference in the communities based on all these sorts of different site level factors. So I measured some things at the photo level and some things at the site level, so I just want to give you sort of an overview. This is a very typical picture that I would take, much less cherry-picked than the last 6 photos to show you some really pretty ones. Has a few corals here, an undescribed sea anemone right here and here. Hopefully we'll be submitting a paper in the next month or so. We'll have a new anemone around these parts. I guess it's always been there. But now we know about it. For each of these photographs, I measured depth with a dive computer. That is 39.5 meters deep. Substratum slope to get sort of horizontal, sloping, or vertical, and those were grouped into those groupings. If you want to talk about that, we can. And then substratum aspect, which is sort of a compass reading. And then all 3 of these plus latitude allows me to calculate a relative annual light index. And so for each photograph, I have an estimate of how much light relative to the surface each photograph was getting. Emphasis on the estimate, but generalizable patterns. Additionally, we had some site level measurements. So at each site, I deployed 2 cinder blocks attached to a brick, and on each of those bricks, there was an alabaster block that when it dissolves, it'll dissolve over time faster if there's more mass transfer at the site, which is relative to water flow given or flow speed if you are comparing it within similar types of environments, similar sorts of flows, turbulence versus laminar flows. Also, all of them had temperature loggers. So each of these sites had 2, one at 10 meters and one at 24 meters. And then these were averaged to get a site level temperature or mass transfer. And then predation pressure. And the way I did this was a catch per unit effort measurement where on every dive, every time, so I knew I wanted to do something with Dermasterias because Ken did stuff with Dermasterias and it was cool and I wanted to do it as well. So every dive, I recorded how many Dermasterias I saw, how big they were. Well, that's not gonna come up in this study. How many there were and how long I was on the dive because I could take dive logs. And so I have a number of stars per hour of diving, and that is what I used as sort of a metric for how many sea stars were at each site. So that's about 150 hours of diving, same thing. Although not always exclusively looking for Dermasterias, but if ever I saw one. And so just to sort of summarize, I had 3 photo level measurements and 3 site level measurements. And the way I analyzed these data was using Permanovas, and every time site was incorporated in, well, in all of the models actually, site was a substratum. The trouble is all of the site level characteristics are correlated, depth and light are correlated, and so you can't just throw them all in one model and tell me how it works. Not to mention, you probably get some horrid six-way interaction my brain around. So I chose particular models that seemed reasonable. I took either the red or the blue group and then added one of the site level characteristics for 6 different models. Then I did depth and slope together and then by themselves, and then light by themselves, and that totals up to 10 if I did my math right. And so I ran these 10 different models, saw which one performed best, and then used that model to explain the distribution of that species. With the idea being that if a bunch of models all sort of behaved, if all of the anemones were reliant on a particular, or one model explained the distribution of anemones better, that sort of indicates a pattern across the Hexacorallians. And so I'll show you sort of each of the models that work best. Each of those on the left is the model, so depth by slope by flow means a full interaction between depth, slope, and flow. Some of them are individual main effects. The blue box is the one that performed the best for each species. Now I'm going to put all the species up, and you can see that there are some patterns here. One in particular is that flow is in nearly every one of these. Flow is modeled as a necessary thing to model in nearly every one of these species, along with depth and light, although nearly every model had depth and light as well, so hard to say. The other thing to mention is that predation pressure and temperature are never models that perform better than other models. Now we're going to get sort of look at the depth distribution of these anemones, because you can glean some information just from looking at these patterns. Before we get into all 7, I just want to orient you to the figures. In particular, I'm just going to keep showing you Ecology because I like them and because they're easy to talk about. On the x-axis, you have density, and on the y-axis, you have depth. You're going to have a little picture of each of the species just for fun, because I like cool pictures of anemones. And what you should be able to glean from this figure is that Ecology is an intertidal anemone. This isn't surprising. This has been described everywhere, but it's nice to see that that works in my study. But that sort of is opposite of all of the other species. So if you look at all 7 species together, I would not try and look at any one species in particular to try to glean something. But if you look at the broad patterns, all but one species, most of the species increase in abundance as you get deeper. In particular, after you get below 12 meters of depth, they start to really build. Epizoanthus and Urdicina don't seem to have a pattern here, but most of them increase. So deeper means more anemones. If you look at how substratum affects these species, horizontal, sloping, and vertical surfaces, and then Y is density, you can see that Anthopleura prefers horizontal surfaces. Once again, Anthopleura is a weirdo and doesn't behave like all the other anemones. Most anemones prefer vertical surfaces, except for Cribronopsis, and once again, Urdicina, which just doesn't care. Or seemingly doesn't prefer something, at the least. I guess I can't tell whether it cares. To more sort of look deeply at some individual species that show are representative of all of them, because I just don't have time to talk about all 7 species. If you look at Balanophylla elegans, which is more abundant as you get deeper and as the substratum gets steeper, there's more Balanophylla at sites with higher flow. So on the x-axis you have flow, and on the y you have density, and there is a relationship between these 2 factors. If you look at Anthopleura elegantissima, so before we even talk about Anthopleura anymore, we're going to see a bunch of these figures. Each of these boxes is a combination of 2 different factors, some of which, so both of these factors actually were measured continuously, but I blocked them so that I could make a heat map out of it. Red means that it's very high, blue means that it's very low, and white means that it's the 40th percentile, just because then you can see the distribution at low values. And you can see that Anthopleura prefers environments with high flow speed, which also incidentally seem to have high fetch. Although I didn't end up modeling fetch, and probably will end up modeling fetch in the long run. But so it goes. And on the y-axis, we have incident light. And this makes sense for Anthopleura. It's an anemone that contains algal cells within its own cells. These algal cells as a trade for more available nitrogen, more available carbon dioxide, and probably not getting munched by copepods. In return, they provide the anemone with lipids and carbohydrates. And so it behooves an Anthopleura elegantissima to live in an environment with high light, but they still need to eat, and so a high flow, high fetch environment is more likely to provide that anemone with the food that they need. Skip that slide. Okay. Metridium farcimen has an interactive response to depth and slope. Good old Metridium. You can see this is a pattern that was present in a lot of these sea anemones. In the initial sort of shallow depths, they're just not present. As you get deeper into middle depths, you end up having anemones on vertical surfaces. And then as you get even deeper, they start appearing on all surfaces. And that was, I think, 4 or 5 of the species sort of show that pattern. Preference for vertical surfaces and then expanding to all surfaces as you get deeper. And we talked a lot about or at least I've hinted a lot at why this is likely the case. If you look at a typical 9-meter depth horizontal surface, it looks like this. Completely and utterly dominated by upright kelps and other filamentous reds and things of that nature. And if you're a 5-foot Metridium farcimen or a 3-inch Metridium farcimen trying to make it in your world, if you're continually getting whomped by kelps, you're not going to do so well. To show sort of more in a quantitative way, here's the distribution of kelps or upright algae. Instead of density of Metridium or density of an anemone, you're now looking at algal cover, and the red being high algal cover, blue being low algal cover. You can see that horizontal and sloping surfaces in the shallows are dominated by these upright algae. And then if you add the comparison between that and Metridium farcimen, it's quite clear, at least to me, that they seem to have opposite distributions. Not perfect, but generally quite opposite. And this hypothesis of algal whiplash isn't a new one. Of course, Paul Dayton already came up with it. He was looking at it a long time ago with barnacles and algae, and a lot of other people have looked at it as well. And a lot of interalgal competition talking about algal whiplash. It seems like it's a significant, it could be a significant contributor to this. And if I had stayed here another year, this is what I would have been focusing on for next summer, was looking at algal whiplash, because I think it has some real potential. Okay, to summarize chapter 3, our final chapter. Predation pressure and temperature, if you remember, had little to no effect compared to other factors, which was surprising given the amount of literature on predation and its effect on the distribution of sea anemones. Depth and light affected nearly all focal species, and this was likely because, 1, competition with upright algae, but 2, that there's endosymbiotic microalgae in one of the species. And so light and depth have different, you desire light if you have, or you prefer light, I guess, if you have microalgae within your cells, but you don't, you want to avoid getting whacked by kelps. And then flow affected most focal species, except for Epizoanthus, which is an oddball we don't get to talk about because we don't have time. This is probably due to more availability of food, but also the addition of a bunch of microhabitats. Just because an anemone lives at a high flow site doesn't mean that it particularly lives in a high flow microenvironment. It could be all tucked into ledges. For example, that undescribed sea anemone that I keep mentioning lives in cracks and crevices on ledges, and therefore probably doesn't experience the same sort of flow regime as Metridium farcimen, who's, I don't know, a meter off the surface. And so where you live in that high flow environment can have some significant impacts on where you are. Not to mention they can just sort of modify that environment by just closing up when it's not fun to be in that high flow environment. So just overarching sort of conclusions of my dissertation, surprising that I can fit it all on one slide, is that I described a new method for marking soft-bodied invertebrates and showed that a stain, some stains can last a year, others are not great to use at all. That's at sort of the individual sort of level. At the more broader population level, you have prey selectivity occurring in Metridium farcimen, being more similar to 80 micron, but also significantly different. And that there was only underrepresented species, no overrepresented species. And that, what was I going to say here? I described a distribution of 7 of these focal species. And the big takeaways from that are that algae excludes anemones in general, but flow increases their diversity. With that, I'd like to thank you guys for being here, listening to me talk on the internet, listening to me talk later on the internet. But before you go, I have a lot of people to acknowledge. There's so many people that helped me. These are my committee, Megan Detier, Jennifer Sink, and Danny Grimbaum. I pulled these off the internet. I tried to get ones that were at least not unflattering. Megan for being there when I'm tearing my hair out, maybe my head, tearing my hair out, just giving me general sort of critiques, reading my dissertation 10 million times at all sorts of phases, and just being like a good second mentor when Ken was away in Italy or something like that. Being around is really helpful. Jen for just being able to, it's wild how good you are at, like I explain something to you and you just take 15 seconds and then explain it back to me in a way that just blows my mind at how broad it is and how, wow, I thought that this was like a really teeny project and you've sort of blown it out into something that makes it more broadly applicable. And also just for tearing my methods and results sections to threads. It's always nice to get someone that does that. And then Danny for making sure that these 3 aren't beating up on me, but also for our sort of conversations, both zooplankton related, but also sort of just where are you going next, what are you thinking about next, and sort of just always having me plan for the next step. And then most importantly, not to insult you 3, is my advisor, Ken, who, amongst many things, was my mentor, dive partner, friend, dive tender, by far, one of his, it was always fun having our lab meetings on a boat as we drove off for me to jump in the water. I'm going to thank a lot of people in the next few slides, and if for any reason I forgot you on these slides, it's not because that I don't appreciate what you've done for me. It's more that my brain is bone at this point, and so I'm sorry in advance. There's a lot of graduate students and previous graduate students and undergraduates and all sorts of things that have helped me along the way in discussions, in particular the Stevens Lab, the Rusink Lab, and the Carrington Lab, which is sort of the trifecta of marine ecology here, as well as a bunch of sort of FHL grads that have been in and out. The FHL Plus community, so all of the staff that are there, I couldn't do half of the stuff I do without their support, and sort of just, I don't know, there's just a lot of technical things about doing a dissertation that, as you guys, a lot of you guys are aware, you can't do without these people. And then sort of a lot of mentorship from a lot of people that were at the labs, either sort of in some time or just generally all of the time, that have been really helpful. And the list of so many people that have dove with me. I've done 138 dives for my dissertation, about 250 something dives for the University of Washington, and I've had 52 different dive partners for my defense. Not my defense, but for my dissertation, which is just wild. There's going to be repeats on all these lists because lots of these people are involved in all sorts of things. 16 different tenders. I had so many students helping me on so many different projects, ranging from measuring corals to measuring soft corals to measuring swimming times of scallops to measuring algal cover to measuring how much hard bottom there is in these photographs. All sorts of things that I haven't talked about too. Measuring, oh shit, I know a few people that I missed already. Tio and there's just too many people to mention. I'm sorry, I'm just not going to do it. But to measuring Dermasterius's interactions with sea stars, just the list goes on, and I wouldn't be able to do. Well, I wouldn't be able to dive without these people because they don't allow me. And then, as is tradition in the Stevens Lab, you have to have a figure that is data, but it's not dissertation data per se. And so I made a figure of all, and this is in line with Kevin Turner made a similar figure, if not the same one, because I'm not as creative as that man, with number of dives or tens on the x-axis, and on the y-axis we have divers and tenders. This is excluding all of the people that only dove with me once or tended for me once, and that's not to reduce the work you've done. It's so that you can actually read people's names, because it would be 3 times as long. Ken by far took the bread with this so much that I actually had to do it on a log 2 scale. And so I think that's 58 tens out of, I think, 98 or so total boat dives. So it's just an amazing amount. And then Sarah and Julia were, in their times, like amazing dive partners, an extension of my own body in a way. And then Mo sort of, although it doesn't show it, dove and tended about the same amount of time, such as the way with a log scale. And then my funding. UW Biology gave me all sorts of money to play around in the San Juan Islands, particularly Bob Payne's fellowship. UW FHL gave me money once again to just continue to play, continue to, I couldn't even list all the different fellowships I've got from there. You know, Megan May's and Richard's fellowship, Ken's fellowship. The list would go on and on. I think I've got about a dozen different ones from there at one time or another. And then the American Museum of Natural History, Larry Gray, during the first year of my tenure here, which let me do a bunch of silly things like the giant cages that I put sea anemones and sea stars in and watch them every several days. which I didn't talk about. But regardless, all sorts of funding agencies. With that, I'm done. Thank you.